So since December, AustinPx has signed up 15 feasibility projects. So the premise of opening the door and see what comes in seems to be working. That's pretty good. But now, so I guess Dr. Miller, other people do agree with you too, that that this result has KinetiSol has a place. But then what? Now we get into the next project, next speak on scale up. And we try to figure out what do we do when those feasibilities work out and they come back and want to do larger volumes. So Dr. Justin Keene, the Senior Vice President of Operations at AustinPx, will present that. So with real world applications and how the process changes, go to make sure we still have the highest quality drug product. Dr. Keene has over 2 decades of pharmaceutical product development. He's a graduate in chemical engineering from the University of Texas. He started his journey as a formulation scientist at DPT laboratories. So he's worked on products like Imodium AD, Tylenol cold and flu, Cool Burst. And then he spent some time over at PharmaForm, where he got into hot melt extrusion and developed projects there too. He's played a pivotal role in the early phase clinical manufacturing on Novafil delayed release tablets. He has a PhD in pharmaceutics. And with that, let's give a warm welcome to Dr. Keene.
All right, well, thank you. So, we get asked a lot, how do we scale up the technology? And usually that conversation starts about when the pictogram that Dave put up earlier gets put on the screen. And so, I think we're all a little tired after lunch and listening to the last talk, but I want to make a user memory. Maybe it'll wake you up a bit. But do you guys picture the pictogram for me? It's a circle, and there's another circle inside it, and some blades coming off of it, and then a bunch of arrows. And so that's more or less what most people associate with KinetiSol. A nice pictogram sideways cut out of the diagram of the system, and some arrows where we're talking about it. And you start talking about blender scale up and mixer scale ups, and we really look forward to tomorrow at Chris's shop so you guys can see this in action. But I'm going to walk through a different set of drawings today to try to get at this picture. How do we scale this up? Because it's really simple, but only after you've had the benefit of Dana's talk and you understand what's important to a dispersion, and even some of the things that were discussed by Josh. Going through the formulation design process, these iterations allow us to start identifying which parameters are relevant. One of the things we get to do in chemical engineering is draw lines through parameters and say this parameter is not important, this is what is important. So before we get into the details, I think it's important to talk about the regulatory considerations, and then we can talk a little bit about these principles and which ones we think are important to the process. Okay, good. So just at a high level, the development objectives from a scale-up standpoint are phase specific. And Dave mentioned earlier, after this formulator scale KinetiSol machine became available, we realized that we can do the whole drug development process of service by taking the knowledge we gain in formulation development and quickly just translating that over to a clinical manufacturing objective. And then we do this scale-up event. And then after that, we do a mode change. And then as we look to commercial demand forecasts, we can sort through how fast we need the machines to operate, maybe how many machines we would need to operate in parallel to meet the demand forecast. So what do I mean by mode? In context of a unit operation perspective, KinetiSol, as we talk about it routinely, especially with the pictogram, is just this little unit operation here. But before that, we need to blend the formulation to feed the machine, and then to make a drug product intermediate suitable for downstream processing, we need to mill it. And so we have these feed unit operations that are compulsory. We can't just operate the machine all by itself. We need to prepare the blend that we send to the machine. And then we also need to post-process it. And we talk about the mode being really just, this is an automation question. How does the operator or the scientist interact with the machine in order to achieve the desired result? And, back to this regulatory perspective, we heard earlier about critical quality attributes. This is part of a broader framework that's well defined in the ICH documents, this Q8, the pharmaceutical development section of our governing, advisory framework about how to develop a drug. And so the first thing we do is define this quality target product profile. We identify what's a critical quality attribute. And then this is where the process engineering piece comes in, is really executing studies to understand how the environment, the materials that are at play, which we already understand a little bit from the formulation development process, and other parameters or attributes of the equipment might affect those parameters and those attributes. And then we ultimately are after a design space for the process so that we can define a control strategy for the regulators. And so in the context of a typical KinetiSol solid dispersion run, it's pretty simple. It can be more complicated, but by the time we're talking about scaling it up, we have a good understanding of most of the variables that are at play. I think Dana mentioned it several times early on that as you develop a formulation, if something from a dissolution or performance standpoint isn't working in your favor, you might want to go back and reformulate. And so this is totally baked into the process we use at Austin Pharmaceutics. Dave and the scientists that work with him produce formulations with the technology, directly with the technology. And so from a practical standpoint, we know that most of the variables around heterogeneity, potency, moisture content, these type of variables we have well under control. So we would expect to see crystallinity as a CQA, and on occasion, if the drug is sensitive, process degradants as a CQA. In the context of scale-up objectives, we're normally asked to scale up first and foremost to deliver some clinical trial material. This is, part of the process. It's built into all of our interactions with our customers. And so we need to run our experiments and inform the control strategy, maybe run some engineering batches in order to accomplish this. And in terms of the FDA guidance that's relevant here, we have this SUPAC term, which I think looking to the future at what we may need to change or understand about the process in order to make a processing change is really important. So we have this scale up and post-approval guidance. And it was revised in 2014 by the agency. And they call out in here why they made these revisions. Specifically, the 1998 version, I believe, came with an equipment list. And the FDA was saying, these are the types of equipments that we classify this way. They operate on these principles. But what industry heard, at least in some instances, was, I need to use some equipment that's on this list. And then the FDA will let me change these parameters and my life will be simpler. And they expressly say, look, this was misinterpreted. And I think it's not just a reflection of this misinterpretation. I think there's a whole aspect of melt extrusion and spray drying becoming part of a common, review pathway for these regulators, and trying to understand the difficulty and complexity of this equipment and bake that back into the guidance. And so we can step through this guidance and really get a flavor for what might be important to a scale-up process. I think if you read the guidance, it's a dry read, but it's got this outline flavor to it. You can step through it and say, okay, well, what does this mean? Why am I even talking about this? It's kind of boring. But these first three are relevant to KinetiSol. We need to blend in order to run KinetiSol. We need to mill in order to run KinetiSol. But we're not performing the unit dose, and I think that's important. There's a whole aspect of what it means when the equipment's creating a unit dose. But I've highlighted this granulation piece here because what's notable in terms of unit operations, what's not on the list is spray drying. What's not on the list is hot melt extrusion. These are captured in this granulation step. And if you dig into the granulation section of the guidance, which again is in outline form, it lists all these types of granulations. Here we find the term spray dry, we term melt extrusion, which is separate from hot melt granulation. So the vernacular gets a little muddy, but we can learn quite a bit more if we dig into these 4 particular operating principles of equipment. Specifically, if you try to make sense of the guidance, honestly, I feel like hopefully there's nobody that wrote this document in the room, right? So I read this and I get to the part where how do you categorize a melt extruder? And I think they just gave up and said, look, this stuff is complicated, and it's a granulator. But that's not true of this other piece, dry granulator, because it can slug or roller compact. These are subclasses. So SUPAC says, here's how we assess equipment, how we assess unit operations for what's important when you change things. And so if you can design your process at the beginning so that it's easy to change later, you can see the whole map. And what subclass is it? What operating principle do we have? And then you get into this whole muddy area. I'd love to have some conversations with folks that have dealt with this in spray drying and melt extrusion. This guidance isn't the end all be all, but it is teaching us something that at least if this document is the only thing you're going off of, HME is a granulator with continuous unit operations. And I think we know that's true. It conveys material to some mixing section. And if we're using the term granulation, this is where that granulation part happens. It's also a pump. There's a vent. This is a very important process. If you don't control moisture removal or residual solvents from an extrusion process, the whole system can go haywire. And then, the term extrusion comes from this last little inch of the machine, maybe half inch. But melt extrusion is a good comparator for KinetiSol because they're both fusion-based ASD forming machines. I pulled this research from my graduate days. I was lucky enough to be sponsored by Roche and Lystritz to do this work on a commercial scale machine at the recommendation and the funding of Dr. D'Annunzio, who will talk next. But we were interested in how, if we configure this machine differently, will it affect the kinetics of the system? Can we manipulate the thermodynamics of the system by changing these machines? And there's a lot of good science to be done here. There's quite a few products made by this process, but a couple things that you can point out here from a kinetic standpoint and from a unit operation standpoint is there's these potentials for long residence time tails. And so one of the things when you think about scaling up and doing high throughput processing with KinetiSol is there's not really a residence time distribution. What we do is we break the blend up into pieces, and then we apply a specific timed energy event to it, which is very short, and then we just repeat that over and over again. And so we don't have this disparity between the material that came out here and this material that might still be in the machine 3, 4, or 5 minutes later. This is a significant potential for drug degradation, one of the reasons why extrusion can be limiting. But the other is a practical standpoint. Dr. Miller explained earlier that we can produce these dispersions at a temperature that's lower than the temperature of critical temperature, I think was the term I learned today. There's a kinetic component and a thermodynamic component here. And what we do with KinetiSol is exploit both of these in just the right way, that it enables fusion processing for a number of drugs, and it's simple. We can make it complicated if we want to, but it is simple. So I'm not done talking about granulation. And the title here is the humdrum of slugging. Is I would expect most of you have heard the term if you've been around pharmaceutical unit operations. Nobody thinks twice about it. You can take a tablet press, which is usually used to make a unit dose, and you can make slugs. And this looks a lot like the KinetiSol batch mode diagram that I showed earlier. We would blend some material, put it through a tablet press, and then mill the material. And now we have a granulation, which we could take to another unit dosage form generating machine, right? And so, there's a question that gets asked a lot. And I think this is a really sort of key question for this process as well. So nobody gets excited about slugging, but what we do is we take powder and we send it through a feeder. That material gets aliquoted into small bits, and then we put energy into that material, and then we take it out of the machine. And it's also a great way to bring back this mode change we were talking about before. I say that word in meetings. Chris says that word when we're trying to talk about the process in front of the circle pictogram, and folks don't really truly appreciate what it means. And I think this is a really good way to paint that picture. So this is a batch mode tablet press, if you will. And if you were developing a pretty simple drug that you wanted to granulate by dry granulation, you could make like a 2-gram slug easily. So I've seen 3-gram slugs on a press like this. You could take that 3 grams of slug, grind it up into granules, mix it with some other excipients, and you might be able to make a couple dozen tablets. You could develop a formulation in batch mode slugging with a tablet press. And so I think what's really the most important part about scaling up KinetiSol for me is the difference between batch and continuous mode tableting. What's important when you make this transition? It's usually to dealing with this system right here. How do we make sure the powder doesn't segregate before it gets into the dye and turn into a unit dosage form? How do we make sure that it flows so the machine can operate at a speed that's suitable for high-speed rotary compression? And the answer is occasionally we can directly compress, but sometimes we need to pay close attention to this feed material to make sure the machine can do what it needs to do. So, I asked the question again, what is slugging? It's a granulator with some continuous unit operations that are happening. So we take powder, we put it in the top of the machine, and the machine on a continuous basis separates out some powder, moves it, works it, ejects it in a couple different ways. These are all, if you were to bound the definition of a unit operation, you could get kind of detailed about this, but things can go wrong when the tablet ejects from the die, like capping or laminating, or maybe it doesn't eject from the tool. There's a lot that could happen here, but in the end, these things are happening. And as pharmaceutical scientists and engineers, we've grown accustomed to the vernacular of this, how this works, what's important for this. And if you understand what's important for making powder flow through a tablet press, then you can scale up KinetiSol. We put powder in the machine, and it does almost the same thing, except for I learned another term I think Dave used earlier, which was KinetiSoling. So I think I need to put that right there. But instead of slugs coming out of the machine, we get shots. That's the term for it here, and it has roots in injection molding. If you're around NuvaRing, you may even hear these kind of terms used, but it's not much more complicated than this. This is a simple process. The way the material flows through the system, I assure you, is simpler than the TIMS. And draw it. So we put the powder in the feeder. The feeder controls the shot size, and we can do this with a high level of control. But what we need to do is make sure it can get into the chamber quickly and reproducibly. And so there's a series of actuators and mechanisms to allow that to happen. And if you want to know why this is at this angle and how this actually works, you're going to see it tomorrow. And that's, it's really cool. It's really simple. But what I have here in orange is the product contact surfaces. These is where the powder goes through the system after it gets processed in this chamber. This is your pictogram turned sideways. This is where the magic happens. And then we take the heat out of it. And so now we can talk about process engineering, because we understand the regulatory perspective here and what's practical in terms of this. This is a continuous process that involves some pre-batching, and what's important here is contained in just about every textbook on the topic. I like this one. It's a good one. But what we need to do is scale the blend first and foremost. And we need to make sure we keep the number of blending events constant, and we match the Freud number. This is standard pharmaceutical art. What we do is take the size and the speed and turn that into a dimensionless number. We do some math, and then I don't know, I prefer to just buy good blenders that all of this is just baked in. And just when it works at 25 RPM on the bench, it runs at 6 RPM on the 300 liter bin, and it just works. This isn't the hard part. What's really we need to understand are all of the things that we've been hearing about today. What's the particle size distribution? Are there density differences that could cause segregation? Are we going to get mass flow? We didn't talk about that today, or funnel flow as the material moves through the system. And so, measuring the wall friction angle with like a shear tester, these are basic things that are known to pharmaceutical engineers that we can collect. And then what we do is we use that information to just understand if there's a segregation mode. If there is, fix it. Is there a mass flow problem? If there is, fix it. Otherwise, the process manages itself. And I think you saw a version of this curve today too. I think we're going to have Dana give her talk every time I have to answer the question about scale up going forward, because basically what happens in the chamber is relatively simple. We turn the mixer on, and over some course of time, the material gets hot enough where the viscoelastic properties of the system change dramatically, and then this viscous heating regime occurs. And once we have this sort of turbulent system in the mixer, this crystalline drug, starts to dissolve by this Noyes-Whitney equation relationship that we've already seen today. And that dissolution process runs to completion, but we can't see that directly. And what we do is we correlate this to some maximum temperature we want the mixer to run to. I think, again, this is T critical, maybe. If I don't know, I liked her presentation a lot. But really, we can't measure the exact temperature of the dispersion directly. You can't do that in extrusion either. This is an apparent temperature. So what we're doing effectively for the process is generating a correlation between this temperature and the dissolution process that's occurring in the system. And so this is what happens inside the chamber after the material has been actuated into the machine. And we can talk about what's happening in the chamber in very complicated ways. I'm going to put a couple equations up here, and then I'm going to simplify them. And I think over the course of the ball game tonight, some of us are going to talk about these equations, and we can talk a lot about these equations. We can turn this K right here into a different number, and then we can turn the diffusivity term in that number into a whole other set of numbers that have viscosities in them. Like there is a ton of art and science behind what's going on here. But from a perspective of the temperature, what matters is this term here, this saturation solubility versus the temperature of the system. So this changes as the material is heating up. And so we need to pick an apparent temperature that gives us the driving force for long enough. And long enough is defined by the mixing speed, which we'll talk a little bit about more. And so we can't just control on the temperature, because sometimes this temperature is too hot. And when it's too hot, we get process degradants. So this is the fundamental, relationships that we care about when we run the mixer. We want to find the right temperature that gives us the driving force for dissolution, but doesn't cause significant degradation. The other parameter that is important is the speed. This is the a speed inherently is like an RPM, right? There's a time factor here, and I learned at DPT a long time ago that we scale up stirrers in a very predictable way. It's not a perfect relationship, but in the end, the tip speed of the machine is relevant. But if you think about what I presented at the very beginning, the first thing we're doing is developing the formulation on a formulator, and then we transition that directly into formulation sort of validation, get into the human PK study and make sure the formulation's behaving the way we want it to behave. And then we do one single scale up. And it's not a large change. I think it's a factor of like 6 or 7. And we can just adjust for that. We just change the speed. We know what speed to set it on based on the speed we ran the last machine. It's not complicated. But where the RPM gets complicated is it does affect this term. I keep adding equations. I apologize, but I could have put a lot of terms here. Really, what you should see here when I talk about this dissolution coefficient is it's a relationship between diffusion and the boundary layer. And I really like the photos I saw. I'm going to talk about Dana's presentation the whole time. She showed a couple really good images where crystals were dissolving. And if you were looking closely, you could see that there was actually as the crystal was dissolving, there was another boundary where the crystal used to be. That was sort of the saturated drug layer. It was a stagnant diffusion layer that the drug has to go through to get out of the system. And one of the best ways to make those go away is with shear. And so we've got this high shear mixer, and so we can make that boundary layer go to almost nothing. In fact, I would argue a lot of the crystals fracture and the area goes up, but really the temperature also affects this, and so does the RPM. So we have 2 different parameters. Both of them make the dissolution rate go faster. One of them, on occasion, can cause the drug to degrade. But there's all kinds of other settings, geometries. We could list 100 factors. We could do that for the tablet press. We could do it for an extruder. We can do it for a spray dryer. We don't need to spend a lot of time on these. We've developed the machine around a practical and pragmatic space. The formulation has made it through the formulation development process. And so we know that it's amenable to the technology. And so when we get to the point where we're scaling it up, all of these parameters, some of them may change, and we can take a page from tablet coding. This is an energy balance that I would construct around a shot basis. And if you're interested in how this works, there's a couple of great articles in the tablet coding space that explains the utility of this type of an engineering approach to a scale-up problem. But there's, effectively, this first law of thermodynamics that says we can't create or lose energy. And so we can say, the conditions before the shot and after the shot the energies that are involved have to be equivalent. And we can organize those energies that are at play into terms. This one is the term that describes how we heat everything up in the system. So you would take the heat capacity, the temperature change, and the amount of material for each of those, and that would be this term. But there's also always moisture. There's always sun, and we're liberating it in the process when we heat it up. And so there's an evaporative cooling effect when we have this moisture term. So moisture is a factor that we need to be aware of. It affects the kinetics. It also affects the total amount of energy we need to put in the system to get the same result. If we add more moisture, we might need to add more energy to the system to get to the same point. And so there's this energy of mixing, which is just when the drug dissolves in the polymer, there's a thermodynamic interaction that may have a positive or a negative sign. And then there's a heat loss parameter associated with the equipment itself. And we can affect that term. And from a process engineering standpoint, we can change the temperature of the cooling fluid that's running through the machine. We can choose the flow rate. But the place we see it the most is as we try to speed the machine up, because after we made our first scale change, the next thing we want to do is switch to a continuous mode. And usually the objective there is to deliver volume, throughput, maximize yields. And so as we start to speed the process up, the starting temperature of the system, this T1 might be different. But again, we know we need to put a certain amount of energy into the mixture to dissolve the drug in the polymer, but we don't want to put too much in the wrong way because that could cause degradation. So there's a balance here. But effectively, as a process engineer, what it means is if there's variation in the environment that affects the heat term for heat loss, if we change the cooling rates, if the moisture content of the system changes, this balance changes. And what we need is a robust process that can adjust to that. And so here's everything. I think I totally violated the equation rule for a presentation, but people are still looking at the screen. So I think it was just the right amount of equations. But in short, what we control is this apparent temperature that we want the material to reach. What we control is the speed of the mixer. It's a mixer, right? Like, how many of you walked into a plant, seen a big vessel, and somebody's pouring salt into water and there's a mixer running? Like, in the end, it's a mixer. There's not that many parameters to get hung up on, because everything else can just roll up into this heat balance, and we can understand that and we can account for that. So here's the non-equation version of this parameter set. We've got the maximum temperature the material may achieve, the speed, the shot mass, some material parameters, some environmental parameters, and then we have some other parameters associated with affecting that heat balance of the system. And what we see typically, and you'd see this graph, I think the Merck team wrote a really good chapter in a book that describes this space for melt extrusion. But basically, if we don't put enough speed or time or temperature on the system, we end up with residual crystallinity. And if we put too much, we might end up with degradation. That's molecule specific. We really like the molecules that don't degrade. So, just bring those on down anytime. But for the most part, the time durations here are so short. They're just so short that we can accommodate this just by the nature of the process. And so what we see is we're controlling the speed and the temperature, and we create a design space where we get the target critical quality attributes out of the system. And then if we change the shot mass, which is an input parameter, it's very easy to control. We're talking about 100 gram, 200 gram shots that are push button control. If we shift the mass, it may affect where we lie or may push us out of the design space. But that's what boundary conditions are for. So we can bound that parameter, we can set that parameter, we can set it and forget it and never touch it. Everything else can also affect where we're at in that design space. And what we're aiming for is a process that's robust enough where this isn't going to push us out of any of our specifications. But really, in practice, if something shifts the process, we can detect that and we can just shift it back. This is an energy balance. Something's affecting the energy of the system, counter it. If there's too much energy being put in, take some away. It's not that hard. We can make it hard if we want to, though. So the machine's really fast in terms of its control and PID loops. Chris will talk about PID loops. There's a programmer in the back here, I think, that probably loves process control parameters. But we had a process where we had observed some segregation, and we needed to deliver a bunch of clinical trial material. And so what we did is we roller compacted the material before we fed it to the machine just to take away that segregation. There's more than one way to do that. We could have gotten different particle size distributions of the materials and not had to roller compact it. But we needed to deliver clinical material. We had the machine. We did that. And somebody said, well, what if the granules affect the process? Well, we just set the machine to run 2 stages and mix it for a little while. We know what mixers do, especially the type of mixer we have. It's just going to grind those particles away. And so we just mixed it at a low energy for a while to remove that granulation history that we added so that the material could get into the machine. And then the process did what you see normally is, starting here, we ramp the RPM up to a speed that affects the viscoelastic property changes in the system that allows for the dissolution process to happen. And you can see, this was a couple hundred shots, and the only real variation is in where this inflection point happens. And that is what we call the sort of when the process kicks off. When you're watching the machines run tomorrow, don't forget to listen, because you can hear this. When the mixer starts to become bogged down by the energy required to keep its motion, the whole viscoelastic nature of the system changes, and you can hear it in the machine. And whether or not that kicks off at, I don't know, what is that, 11.2 seconds and 14 seconds? I mean, it's kind of funny. It's like plus or minus 2 seconds. Like, this is not relevant. What's relevant is that the shape of this curve and the endpoint of that curve and the speed is the same every time. And so this is that energy balance I'm talking about. As we're running the process, maybe it was a little hotter and kicked off earlier. Maybe we stopped the process or the first shot kicked off a little later, but this doesn't affect the quality of the system or the product that we're making. And you can see that in other parameters. Like, if you were to take a data set like this and construct a process control chart, process engineers love process control charts. This is a lower control limit and an upper control limit, but from the purest statistical standpoint, I don't like statisticians. Are there any statisticians in the room? This number is meaningless from a product quality standpoint. What it's telling us is information about this data set. And this is where we can get an understanding of what's happening from this heat balance standpoint. So each one of these dots is a shot. And the first shot is being put into a machine that's got coolant running through it. It's kind of cold. And so it takes a little while for the system to generate up just enough heat for that kickoff to happen. And then the second shot happens a little quicker. And then the system stabilizes. Maybe the operators went to lunch. You know what we do when it's lunchtime at AustinPx? We stop the machine and we go eat lunch. I don't think you can do that with a spray dryer and extruder. It's those little things that make me happy. We stopped the machine, we started it again, it works. And we know it works because we can map it, we can test it. We have all these great scientists sitting around here that have taught us how to do this. One of my favorite scientists is Angela, because She knows how to work our ramen process analyzer that we can take into our GMP suite. And we use this really to map the process. We don't need it for process control standpoint, but we can. And so what we can do, because we're making a shot by shot by shot basis, and you'll see Chris and the team tomorrow, you can just walk up to the machine and put in some parameters and it'll make a shot. And then you can take your Raman shift with your validated reference curve that's been trained. We could have a whole conversation about this, but in the end, we can just map all of the parameters. The 2 parameters we care about, maybe a third. If you really want to get fancy, we can add some moisture, take some moisture away. We can map these 4 parameters and understand the whole system. Like this. So, this was really cool about this is we needed to, I keep saying we needed to deliver some clinical trial material because that tends to be very important in early phase development. I don't think it ever gets not important, but it's always looming. We don't get to just run experiments all the time. Some companies do. I'm not sure where the resource balance is there for you as an individual or at your company, but we can do things like this. It's really cool. This is like the 200 shot. We delivered the clinical trial material quantity we needed to, but we wanted to understand the system a little bit more. And so we just wrote up a little protocol, and at the end of the batch, we just started testing stuff. Let's change the RPM and let's make 3 shots in a row. Because after 3 shots, the energy balance. Change it down, 3 shots, change the shot weight. I mean, this took like 30 minutes. It's not even that. I mean, this is really powerful. I really, as a process engineer, so cool. Is it? Yeah, I mean, yeah. So then we get to the boring part of the process, but it's the part of the process that I think people think there's a lot of risk. So now we have this dispersion. We know how to make it. We know how to make it over and over again. We've demonstrated it many times, but we do have to deal with this hot mass of sort of aerated, rubbery material. I think from a material standpoint, if you go read a material science textbook, it's a rubber until it passes this glass transition temperature. It becomes a glass, right? And so we've got this rubber, and we want it to get past its TG, and it's doing that with this sort of quenched plate system because it's pretty practical. The mass comes out, these 2 plates come together, and they pull the heat out of the system. And you can ask Chris about it tomorrow, but the first thing I did was make him connect the earliest machine we had to this quenching machine. There were 2 separate machines at the time. I made him connect them so that I could measure exactly how long did it take to quench, how long did it take to get to the temperature we needed it to be at. And we looked at it for a couple of years, and then he put all this stuff into the same machine. So all that data is collected all the time, but it's actually become it's irrelevant data. When I mentioned at the beginning, one of the cool things that chemical engineers get to do is just take their pen and draw right through a term and say, that's not important. This is one of those things. And it becomes a very practical matter. Did we get it vitrified? Did we make it into a glass? And then, is it going to melt the polymer liner on your containment that it's coming out in? Is the operator going to touch it? Is it too hot to handle? Beyond that, there's not a lot to it. We're just taking the material out of the system, cooling it to some practical temperature, and we can start to press this time as throughput becomes important to the process, as we care about efficiencies, as we care about yields. I don't care about that much in phase 1. Not that much in phase 2 either. But for commercial, it can become important. So this becomes a practical thing about throughputs and potentially, yield. So what's really important about quenching is that the materials don't stick to this. That's probably a scale-up standpoint as we start to get bigger and hotter and faster, just making sure that we have the right materials of construction on the equipment, and we've studied that. So the other aspect of this is, there are parameters, and they'll show up in our batch records. Maybe for some systems, we might find that they become a critical process parameter. I doubt it. I really do doubt it. But the two parameters are quenching delay and quench time. And so the material leaves the system. It needs to get into a location where we can quench it and take the heat out. And we need to tell the machine how long to do that. And where we set that time is where the cooling curve for the system would diverge. And process engineering wouldn't be complete without an equation. There's a total heat term here that's based on the convection of the system, the area, so the size of the plates, how much force we put on the plates to make the shot squeeze down. And then we can change the coolant. The shot temperature's more or less already set because we set that parameter for the previous part of the process to cause the dissolution process. But we can affect quenching in the absence of making the dispersion by changing the coolant. But the shot mass is also a pretty relevant factor here. But again, it's a practical matter. Our experience set teaches us something. And I think I heard it at least twice today that, if you're formulating a dispersion and your performance is affected in some negative way, or you can't make tablets in some way, maybe you're using the wrong process. Maybe you should go back and reformulate. Every one of you has one of these little shots in front of you that came out of the formulator. And so what's really, I think, important, and not to be, don't dismiss it as a relevant piece of the puzzle here. We formulate with this small machine. It's empirical, it's practical. We can do it rapidly, and you'll see that tomorrow. But what it means is everything that's made it to the point where I need to scale it up is proven to have survived the quenching process, which involves, at the small scale, an operator manually taking the material, putting it in between some plates, and then taking the heat out of it. So if that happens routinely and effectively at the formulation scale, the machine can do it. And so you can look at these, this is important from the regulatory standpoint, because it's not just about what that SUPAC guidance says, it's about the conversations with the regulatory authorities. What's the risk? That's what everybody cares about. And so at a hot temperature, there's no driving force for nucleation because we're above the T critical. I'm going to get this right. I'm going to factor these terms in. But then, when we get it down to low temperature, we vitrify the dispersion. There's no molecular mobility. If you have a risk right here, you have chosen the wrong process, and you should go somewhere else. You should not be here. But this is really, again, we're talking about seconds of exposure. There's a practical perspective here. This is just a low risk process. So then we get the material out of the machine to provide a dispersion that's suitable for a finished dose manufacturing. We have to mill it. So there's a really practical part that I believe heavily in, that is to do a two-stage milling process. The first reason is because we want to be able to control the feed rate to the mill, and having this big disc makes that kind of hard. So we're going to just bring it down to like the centimeter scale. But also, as we play around with the shot size, as we scale up in terms of throughput and speed and maybe the shot mass, we want to be able to remove the geometry dependence from the system so that we don't have to re-optimize our milling process every time we change a parameter in the compounder. And then, more equations. So we got to mill it. This is, again, textbook stuff. If we scale up the mill from our small hammer mill, tabletop hammer mill, to our hammer width 3, or maybe to a future to a hammer width 6 or Fitz mill, we start again this same equation. A lot of the operations here just deal with rotating things. and we match tip speeds. And then we control these parameters that control the particle size distribution. There's a couple people in the crowd that do this every day for a living. So if somebody asks me a question about this, TJ, you have to answer it. So this brings us back to this sort of same perspective on, so what do we need to do to scale up the process? First and foremost, because we're using a small version of the technology to generate the formulation process develops the first iteration of it. And so then we can just reproduce that to deliver clinical trial material so that we can validate the formulation. And then we do a small scale up event. It's less than 10 times in the geometry factor. And there, it generally makes sense to call the spectroscopists in the room and map the process. And we can do that while we're delivering batches. We can map it on the morning of the first batch and then just deliver the material. Or we could do an engineering batch and then map it some more. I mean, we have a level of flexibility that I don't think anybody will really appreciate it until they see it in action. We can just stop the machine. We can just change the parameters because we can. And nobody thinks about it because you're so stuck. We're using a process where you can't. And, you know, it's, for me, it's very profound. And then we need to go fast. And to go fast, we need good powder handling properties. So we either go back to the supply chain, or maybe we granulate, maybe we don't, maybe it's suitable for direct compression. I like HPMCAS MP, that's big particles. I like cohesive APIs that stick to those particles, then we don't have to worry about this. But this is a practical matter for any pharmaceutical unit operation that deals with powders. You solve that problem, map the process out, make registration batches, and then later, as you try to go faster, you have a set of, I think the term is experience, you know, conditions that you've mapped for the process, and you should have a range around those. And as you try to speed the process up, you just shift around in that space. And if you can't do it visually, which operators for the most part can, and we've proven that, you can bring a Raman tool in the room or some other PAT tool. Every 30 seconds to a minute, something comes out of the machine. That's plenty of time to check it, make sure it's good. You can get feedback control if you want. And we've done this a bunch of times. This is the most public example. Dave talked about it earlier, where we did this with etravirine. This is one of those molecules that was bounded because there was a process degradant. You know, a lot of APIs don't do this in the system. I love those, again. If I didn't say that earlier, I like, but we can deal with it. We can just set a boundary, say like, this is a temperature bound. Some formulations don't really have that. And then we control the speed within that bounded temperature. And this was a data set we collected on each batch just to prove that we were in control of the process. And you know, it works. And it's easy. So, you know, because it's just a granulator, but does other discrete unit operations. And, you know, we can't scale it up if we don't scale up the blending process. That's actually, for me, more, I put more thought into that than I do the process itself. And then, you know, we set the temperature and speed ranges. The design space for the process is simple. It's 2 parameters. We can make it more complicated. We have, but you don't have to. And then you bound the other parameters that may make it go out of control.